Hi, Kusma.
Hi, ma'am.
Let us start the program.
Okay. Yes, ma'am.
Let we start the program. My voice is audible.
Yes, ma'am.
Okay.
Yes, ma'am.
Yeah. Good evening to one and all. Sorry for the delay of a few minutes because of the traffic and all. On behalf of the Department of Humanities and Sciences, KPRIT College of Engineering, it gives me immense pleasure to welcome you all to the inaugural session of the ATAL Faculty Development Program on the theme Microplastics as Emerging Pollutants, Analytical and Monitoring Perspectives. We are truly privileged to be guided by the vision and encouragement of the governing body of KPRIT COE, our Honorable Chief Patron Sri Kommur Pratap Reddy Garu, Sri K. Prashant Reddy Sir, and Sri K. Rakesh Reddy Sir, and Srimati K. Divyasri Madam, along with the constant support of our, it is special honor to welcome Professor Uma Madam, Uma Maheshwari Dev Madam, Vice Chairperson of APSCHE, as our guest of honor. So we are sincerely grateful for her presence and the inspiration she brings to this gathering. I would like to acknowledge Professor Jian Zhu, whose goodwill and association with us remain strong even though she could not be here today. This faculty development program is not just an academic event, but a powerful platform for learning, sharing, and collaboration. It unites brilliant minds to deliberate on one of the most pressing environmental challenges of our time, the silent yet growing threat of microplastics. uh, Through meaningful research insights and academic exchange, we aspire to move closer to innovative solutions that safeguard both the environment and the human health. With deep gratitude to our dignitaries, faculty members, and enthusiastic participants, I once again extend a very warm and heartfelt welcome. Let us together make this program not only enriching but also inspiring, paving the way for new ideas and lasting impacts. On this occasion, I want to introduce our director and principal, sir. So, our respected director, Professor Sudhir Prem Kumar, ex-professor of JNTU, ex-VC of Rama University, uh, his vision and guidance, which continues his vision and guidance, which continues to inspire the progress of KPRIT College of Engineering at Gatkesar. It is also my honor to introduce our principal, Dr. K. Srinivas Reddy, who distinguished professor, academician with over 3 decades of experience, a gold medalist from Anna University and PhD holder from JNTU Anantapur. He has served in key academic roles as a director, principal, dean of R&D, and also he authored a number of research publications, books, And also, Dr. Reddy is also a fellow and life member of several professional bodies, reflecting his strong presence in the academic and research community. Today, we are truly honored to have him with us to share his insights about our college and its achievement, and also college vision for the future students. Sir, Dr. K. Srinivas Reddy, sir, please continue your speech.
Is it audible?
Yes, sir. It is audible.
So good evening to one and all. So this is our college KPRIT COE, that is KPRIT College of Engineering. It was established in the year 2010. It is a premier institution of higher learning located on a 22 acres of land. It is located in Katkesar, Hyderabad. With a commitment to academic excellence and holistic development, the college has earned recognition through approvals from AICTE New Delhi, and our college it is affiliated to Osmania University, Hyderabad. And also our college is accredited by NAC, and also our college is UGC autonomous. At present, we are offering two BE programs, that is UG programs, Computer Science and Engineering, Computer Science CSE, AI and ML, Artificial Intelligence and Machine Learning. And also we are running one MBA course, that is PG course. The college provides modern infrastructure with well-equipped laboratories, digital resources, and skill enhancement platforms that encourage our students to engage in hands-on learning, internships, and collaborative projects. Our vision of KPRIT COE is to emerge as a center of excellence in technical education, nurturing innovation, ethical leadership, and societal responsibility. Our mission is to equip students with strong academic foundations, advanced technical skills, and the confidence to address the complex challenges of a rapidly evolving world. Looking to the future, our college KPRIT COE aims to further strengthen its research culture, expand industry collaborations, and continue providing its students with transformative opportunities for career growth and innovation. With a focus on academic rigor, research development, and global relevance, the college strives to inspire the next generation of engineers who will contribute meaningfully to society and the nation. Our college has 20 doctorates and all our PG experienced faculty. This is regarding our college brief profile. I request Saraswati Madam to continue. Madam? Madam? Hello?
Thank you, sir. Thank you for delivering a lecture regarding overview of our college. So we are introducing guest of honor. So by Mr. Ganesh. Ganesh, sir, please continue.
Hello, good evening, everyone. Is it audible to everyone?
Yes, sir.
Yes, sir. Okay. On behalf of the organizing committee, it is a privilege of to extend our heartfelt gratitude to everyone who has made this inaugural session of the ATAL Faculty Development Programming on microplastic as emerging
Ganesh, sir.
pollutant.
Sir.
Yes.
Sir, you are introducing a guest of honor.
Yeah, guest of honor. 1 second.
Please check. Sir, somebody is Kindly switch off video. Ganesh sir, are you there? Ganesh sir.
Oh, hello. Hello.
Yes, sir. Ganesh sir.
One second.
Ganesh sir. Let me introduce myself. I am Dr. M. Saraswati, coordinator of this FDP. So, great evening to everyone. It is my privilege to introduce our esteemed guest of honor, Professor P. Uma Maheshwari Devi, a scholar, researcher, and a leader in higher education whose journey is true source of inspiration with a distinguished academic career in applied microbiology. She earned her PhD and went on the complete a prestigious postdoctoral fellowship at Penn State University, USA. Her pioneering research focuses on natural products and their therapeutic applications, with a special emphasis on marine microorganisms for the treatment of cancer and diabetes. Professor Uma Maheshwari Devi has nurtured generations of researchers, having guided one postdoctoral fellow, 13 PhD scholars who have successfully completed their research, and six more who are currently pursuing theirs. Her remarkable contributions include 5 patents, 92 impactful publications, and more than 3,700 citations reflecting the global relevance of her work. She has successfully undertaken 18 major research projects funded by premier agencies such as UGC, DRDO, DST, and DBT, while also holding several key administrative roles with vision and dedication. Her excellence has recognized with numerous awards and honors. The AP Scientist Award in 2021, the UGC Mid-Career Award in 2020, Fellowship of the AP Academy of Sciences in 2018, the State Best Teacher Award in 2017, and international recognition such as the DBT Crest Fellowship in 2011 and Biocast Fellowship in 2007. Her illustrious journey began with the Young Scientist Award from the Indian Science Congress in 1997, and since then she has continued to inspire through her academic brilliance, innovative research, and unwavering commitments to education. We are truly honored to have Professor Uma Maheshwari Devi with us today. Her remarkable career and visionary leadership remind us of the heights the dedication and passion for science can achieve. Please join me extending a warm and respectable welcome to our guest of honor. Please join us, Madam.
Thank you, Saraswati, for a nice introduction. I think I'm audible.
Yeah, ma'am.
Yes, madam.
Yes.
Respected Pratap Reddy Garu, that is the chairman, Prashant Reddy Garu, vice chairman, Sudhir Prem Kumar Garu, very good friend of me, that is a director, Srinivas Reddy Garu, principal, Saraswati, she's contacted me for the last 1 week, and Nagendra, Dr. Nagendra, who is also the he's also co-coordinator, and I had seen that the Chinese Academy of Sciences is there. Jian, I think she also joined as a guest of honor for this opening ceremony or inaugural ceremony. Let me congratulate all the faculty who joined for this faculty development program. I think that it's a honor for me and also duty or responsibility for me to speak on this current issue, the burning issue. I need to congratulate or appreciate the energies of KPRAT College of Engineering for choosing this beautiful topic for faculty development program and ATAL scheme itself. Definitely, whenever we are looking into the plastic pollution in the social media, whether it is the internet or it may be the TV channels or the papers, we can see that the plastic pollution in the form of plastic bottles floating on the water, or maybe blocking the sewage pipelines, or maybe like that. Maybe during the rainy season, also we can see that that is the plastic pollution. But majority of them, they're unable to see that what is happening beneath this plastic, that is the microplastic, which are very very minimal in size, less than 5 millimeters in size, and how they're impacting the lives, that is slowly they're infiltrating into our ecosystem. Not only into the our ecosystem, it is also leading into the animal system, including the human system. Now today we can see that slowly they are entering into our blood, our placenta, as well as the liver and the lung. That is the impact of these microplastics. So we need to appreciate the KPRIT College of Engineering for taking this burning topic. And even I looked into the syllabus, that is what they framed for this faculty development program. They concentrated equally on the mitigation strategies as well as the analytical tools and monitoring perspective. After looking into that, I got into my mind that there are two things. That one is about they're giving the picture of overall picture of what are these microplastic. Whenever the microplastic, always we are worried about whether they're the primary microplastic which are coming from, that is in the form of the microbeads from the cosmeceutical industry. The that is cosmetic industry, it is playing a prominent role in contributing the microplastics to the either to the oceans or to the rivers or maybe to the soil or maybe the air. We are, that is totally, if you look into the India, majorly we are depending on the cosmetic industry, but they are the major players in contributing the microplastics to the ecosystem itself. In addition to that, microbeads, even the industrial abrasives you can see, and also the synthetic fibers which are coming from that is the textile industry. These are contributing as a primary microplastics, and equally the role played by the secondary microplastic, which are from the everyday, from the home to that of the industry. We are contributing plenty of microplastic into the system, the ecosystem. I can simply say that these microplastics are ubiquitous in nature. Wherever you see, whether it is the ocean or the river or the air, whatever it may be, or the soil or the human intestine, wherever you see, finally the microplastics are loaded. And there is one saying that for every week we are that is we are ingesting 5 mg of the plastic. We are unaware of that, but we are ingesting, we are taking for nearly 5 mg of the microplastic. So what will happen to this microplastic? That is this faculty development program. You need to understand that why it is a that is emerging as a pollutant. So it is emerging. They are given a beautiful title, that emerging as a pollutant. Why? Because these microplastics, they're having the unique feature and the complex feature, and the bioremediation of these microplastics is plastics. Slowly, we are developing the strategies for degrading the plastic, either by means of the green approach or by means of the chemical approach. Whereas this microplastic, because of their small size, because of their tiny size, and the complex diversity of the polymer material, still we are struggling to develop the analytical tools also. Even though the analytical tools like the spectroscopy is there, the scanning electron microscopy is there, LCMS is there, FTIR, even though after the advanced tools are there, still we are struggling, the scientists are struggling for isolation of this, identification of this. That is one strategy we need to address, that how best we can develop, we can make use of the analytical tools, particularly for the isolation and identification and the purification of the microplastic. That is one aspect you can learn from this faculty development program. Then coming to the monitoring perspectives, we are having several approaches for the monitoring, but my intent to appeal to all the participants is, in addition to that, the monitoring approaches by using the analytical tools, you need to look into that what is happening, that the bio monitoring, how they're entering into the animal system. You know that majority of the fish, that is a fish industry, aqua industry, now you can see that a lot of fishes are going away just because of these microplastics. In addition to the fishes and the animal system, Now it is entering into the human system, and one recent publication is there. There is a chance for that is always the microplastics. They can hit at the inflammation, or they may target the oxidate. They may lead to the oxidative stress. If they are targeting the inflammation and the oxidative stress, there is no distance in order to develop into the cancers. Maybe in the future of in couple of days or maybe couple of months, we can link the microplastic with the development of the cancer also. Why? Because now, because of this, you can see that slowly they're causing the inflammation and they're attacking the kidney also. So lot of things are happening, especially for the human system is there. So when they're entering into the human ecosystem, the human system, then there is a lot of danger is there. We need to look into that, how exactly these are hitting the pathways. Several pathways are there that you can learn in the case of the monitoring perspective, especially that is bio monitoring. What are the strategies we can apply for bio monitoring, that you can learn during this faculty development program. As a microbiologist, my concern is the mitigation. There are several mitigation strategies are there. One is reducing the micro pollution, that is a microplastic pollution, that is by avoiding the single use plastic. That is one thing. And the second one is community awareness. We can create the community awareness to stop the plastic uses. At least the single uses plastics we can control. And the third one is we can introduce the education, that is the plastic free education we can do. And in the academic institutions, we can totally make use of that is the plastic alternatives instead of using the plastic, but totally each and every day from the morning, from the that is after awakening, we started we are starting use of this plastic, so that dividing or avoiding the plastic, it is a very great problem. I know that majority of the people, even the policies, lot of policies are there. And recently the UN they had given a call that we need to think of that what is this mitigation strategies for the microplastic. But as a microbiologist, my concern is that because in even your FDP, majority of the classes are focused on that is bioremediation strategies. So these bioremediation strategies, where exactly we can go for the bacteria or the fungi. Now we are using the nanotechnology, also the nano advanced materials for the bioremediation of the microplastics. So this is a very good approach in order to mitigate the problems of the microplastic by using the different approaches, that is a bacterial and fungal, as well as the nano that is a photocatalytic along with the nano catalyst also. And the recent methods are also there, that is engineered enzymes are available. Now slowly we are because already the Chinese Academy of Sciences, they partnered with this KPRAT college itself. So all the faculty who are attending this faculty development program, try to take it as a serious faculty development program and try to learn that how best we can solve this problem, because it is not a it cannot be only the that is a government duty to take care of this. It is a combined action of academicians, industry people, as well as the policy makers. When three integrate, they can think about this, then only the problem, the solution will come. But as a faculty, as academicians, you need to carry out the research, and the research must be, you have to be in a focused manner. That is, the majority of them are the biology or their engineering or their, so that the biology people always shake your hands with the engineering people. Try to design some of the things, that is maybe you can modify the enzymes, that is by using the genetic engineering methods, or maybe you can use the CRISPR-Cas technology for engineering the bacteria, or you can design that is AI-based tools for imaging how best this the microplastic are entering into the liver system or the lung system. You can develop the computer scientist, you can develop the image analysis. So with the combined effort, there is a possibility as well as the feasibility to mitigate the microplastics. Then only I think this faculty development program is successful. Anyhow, I congratulate the KPRAT college, and I can request all the academicians who joined along with this group, and you can you will become the leaders and you are the torch bearers in order to look for the strategies how best we can control the microplastic pollution, and the emerging pollutants must be maybe we can say that the emerging pollutants may be divided from our ecosystem, at least from the human ecosystem. Thank you very much for giving me this beautiful opportunity.
Thank you, ma'am. Thank you for wonderful speech. So, thanks, Mr. Satish. Satish, sir. Sir. So, thank you. I think this is so on behalf of the organization committee, it is my privilege to extend our heartfelt gratitude to everyone who has made this inaugural session of the ATAL Faculty Development Program and Microplastics and Emerging Pollutants, Analytical and Monitoring Perspectives a memorable success. We are deeply thankful to our chief patrons and patrons for their constant encouragement and vision. A very special words of gratitude goes to our esteemed guest of honor, Professor P. Uma Maheshwari Devi, for gracing us with her inspiring presence and for reminding us of the power of the science to transform lives and societies. We are sincerely thanked to our honorable governing body of KPRIT and the principal K. Srinivas Reddy sir, whose leadership and guidance continue to shape the academic and research culture of our institution. We are also thankful to all our respected resource persons for bringing their expertise and Sting donor. Sorry, wait. Sorry for the inconvenience. అది మ్యూట్ ఎందుకైంది? ఎవరు చేశారు మ్యూట్?
మ్యూట్ లేదు కదా. హలో,
Ma'am, you
mute
are audible now.
Okay, right.
Thank you. So let us finish the inauguration with a delay of 5 minutes. Sorry. With collective efforts, may we not only deepen our understanding of microplastics as emerging pollutants, but also contribute towards building a healthier and more sustainable future. Thank you once again, and we wish the program great success in the days to come. Thank you, one and all. So let we take 2-3 minutes. Again, we will come back. దీంతో జాయిన్ అవ్వాలి సార్. ఇది పక్కన
దాంట్లో చేయండి. అదే నెట్ కనెక్ట్ చేసుకుంటాను. చూడండి
ఎవరు రాలేదు చూడండి సరిగ్గా చెప్పడానికి.
అందరూ వచ్చారు.
మన వాళ్ళు వచ్చినారు కానీ చెప్పలేదు. నేను చెప్పుకున్నాను సార్. Let us start the session again, the first session. So I'm going to introduce Dr. Emmanuel Vijaypal Panditi. Good evening, respected dignitaries and distinguished guests and dear participants. It gives me immense pleasure to introduce our keynote speaker for the first day of this ATAL online FDP program on microplastics as emerging pollutants, analytical and monitoring perspectives. Today we have with us a scientist whose journey is a true inspiration to both aspiring researchers and established professionals, Dr. Emmanuel Vijaypal Panditi. Dr. Panditi is currently the head of the Microbiology and Biocatalysis at Zanoprima India Private Limited, Hyderabad, where he leads cutting-edge projects on enzyme discovery and application. His pioneering work focuses on eco-friendly enzymes capable of degrading xenobiotics and microplastics, a breakthrough area in the fight against environmental pollution. He earned his PhD in biotechnology from the University of Hyderabad, where the engineered microbial systems for the degradation of toxic pollutants. He further enriched his expertise with international exposure as a visiting scientist at the University of Texas Health Science Center, USA, and as a research scholar at Texas A&M University. His academic brilliance is complemented by industrial leadership, having built and guided R&D teams at Aurobindo Pharma, Albus Ecogymes, and Zanoprima, India. Dr. Panditi's contribution include over 15 impactful research publications in high-quality journals, innovative enzyme libraries for biocatalysis and biodegradation, and successful scale-up of enzyme production processes to industrial pilot plants. His work not only advances science but also offers practical solutions to real-world environmental challenges. What makes Dr. Panditi truly remarkable in his vision, the belief that enzymes, nature's green catalyst, can be harnessed to heal our polluted ecosystem and create a sustainable future. Ladies and gentlemen, please join us in warmly welcoming Dr. Emmanuel Vijaypal Panditi, a visionary leader in the field of bioremediation and environmental biotechnology. Sir, good evening, sir. We are welcoming you for your session.
Good evening, everyone. I thank the management of the college and Dr. Saraswati Garu and the principal of the college and the committee of the faculty program for inviting me to uh, deliver this address.
Welcome, sir. Welcome.
I think you can all see the presentation on the screen.
Yes, sir. Yes.
So the topic that is given is role of cold adopted enzymes in microplastic degradation. So I just want to give a very brief introduction so that uh, some of the students who have might have joined, it will be helpful for them. So plastics are synthetic polymers which are specially derived from petroleum and are widely used in industry and domestic applications and are considered very important for our present day life. So, what are the key characteristics of synthetic polymers? These polymers, which we use regularly, are very lightweight, they're stable, they're chemical, and their physical properties are very strong, they are durable, and they are very easily produced in an industry. In 2016, the whole world has produced around 335 million tons of these plastics. In 2019, we produced something around 430 million tons of plastics. But there is a small dip because of the efforts of various organizations. In 2023, um, the Overall, synthetic plastic production has reduced from 430 to 400 in 2023. So, why are these plastics so uh, recalcitrant? So, it is because they have a backbone which is like a carbon-carbon backbone or a carbon backbone with a heteroatom. So some of the examples of plastics with a carbon-carbon backbone or polyethylene, polystyrene, polypropylene, and polyvinyl chloride. And plastics with heteroatoms in their backbone or polyethylene terephthalate, which we generally call as PET, and polyurethane. So plastics, when they are produced, we produced in two primary forms. One, one are very small in nature, and we call them as microplastics, and they are called as primary microplastics because the purpose we have people produce is they are used in various industries, and they want them in a very minute nature, less than 5 millimeter in size. But large plastic products generally degrade and release small micro particles, and they're called as secondary micro uh, plastics. And these microplastics and sometimes the nano sized particles, they enter into our terrestrial and aquatic ecosystems, and where they accumulate and cause a lot of environmental pollution. As I have told you earlier, so there are primary microplastics and there are secondary microplastics, and they form a different, they come in different shapes like forms, fibers, beads, fragments, and the backbone you can see they can be of ethylene, polyvinyl chlorine, or polypropylene, styrene, etc. So, what is the environmental impact because of this microplastic accumulation? These microplastics are very much recalcitrant and they are very hard to uh, biodegrade in the environment, either in terrestrial or in aquatic system. They have a long duration for degrade, or they have a very long half life. Usually, some of the plastics take even more than 600 years to degrade. Natural degradation can take up to 5 to 100 years for PET degradation, and it may take more if the temperatures are low and if the oxygen is limited. The pollution, as our chief guest has already informed, that it is a major global problem, and even human milk, it is reported human milk also have some microplastics. So why is that is a cause of concern? Because not only microplastics uh, are deleterious to the health, but they also have the tendency to accumulate or attract toxic compounds such as bisphenols, polybrominated diphenyl ethers, and polychlorinated diphenyls. So, what are the general challenges for degradation of the carbon backbone plastics? Unlike hydrolysis susceptible plastics, but as the biodegradable plastics, the degradation of polymers with a carbon carbon backbone is primarily initiated by UV radiation and oxygen. This photo initiated oxidative degradation leads to fragmentation or chain scission, reducing the molecular weight before subsequent biodegradation can occur. The carbon carbon bonds are highly resistant to enzymatic cleavage. Currently, the mechanism for degradation of the covalent bonds of the carbon backbone in the petropolymers is very less known. So let us focus on some of the individual polyplastics, polypetroleum plastics. First one we can see is polypropylene. It is a very inert material, very long chain molecule with methyl groups. There are very few reports on biodegradation of polypropylene, but there are certain bacteria such as Pseudomonas and Vibrio and fungi like Aspergillus niger. When
Speaker ఇక్కడ ఆఫ్ ఉంది
these plastics are given in in the feed of these bacteria or fungi, they are able to co-metabolize but not completely degrade this polymer. Coming to polyvinyl chloride. Polyvinyl chloride is a very rigid plastic which we regularly use in household, and not only polyvinyl chloride is mixed with several additives which make them more recalcitrant. But there are certain lignin degrading fungi or mushrooms which can degrade PVC in a very small amounts. And certain bacteria like Pseudomonas citronellosis and Bacillus fluxus, and some of the insects, Tinobria molitor, in the form of their larvae, can depolymerize PVC and mineralize in a limited quantity. When coming to polyethylene, just like polypropylene, polyethylene is also extremely resistant to biodegradation, but there are certain bacteria which are known, and there are certain insects in their larval form which are known in the recent years that they can assimilate polyethylene. Bacteria like Bacillus, Rhodococcus, Pseudomonas, they can degrade UV or thermal pre-treated plastic because of the alkane hydroxylase system which is present in their system. The polyethylene breakdown will usually give ethylene glycol or ethylene, which can be directly used as fatty acid metabolism. The greater wax moth, which is a parasite or which eats honeycomb, the waxy part of the honeycomb, also degrades many plastics. The larval form of this greater wax moth, Galeria melanonella, can eat plastics, but in a limited way. The larvae can eat this plastic, but they can't grow on this plastic. So the feed has to be substituted, and it can co-metabolize the polyethylene plastics. Some of the bacteria which are present in the gut of this larvae might be responsible, along with the enzymes of the larvae itself, for degradation of these plastics. The other polyplastic is polystyrene, which is having a aromatic ring. Because of the aromatic ring, it is very stable. It's a high molecular weight, hydrophobic material, and it is also very much resistant to breakage. As I told you earlier, some of the enzymes produced by the mealworms or wax moths, they are able to metabolize these plastics because of the bacteria which might be present in their gut or the enzymes which they might be produced. In a similar way, some of the fungi like white rot fungi and the mushrooms, they can easily degrade small quantities of these plastics when present in a defragmented form. So there is another class of petroplastics which contain heteroatoms, and they are susceptible to hydrolytic cleavage because of the presence of ester or amine bonds. Polyethylene terephthalate is globally produced because of its very good durability, and it is used in all plasticware, all plastic cookware, and some of the lab plasticware and everything. PET has a high glass transition temperature, but above the glass transition temperature, it can form or it can change from the crystalline or semi-crystalline state to a amorphous region, which becomes more accessible to microbial attack. In 2016, a bacterium called Adenonella sacchensis was isolated, which can depolymerize PET and use the terephthalate subunits for its growth. The way this organism degrades the PET is by producing enzymes like PETase or METase. The first enzyme is PETase, which converts the PET to monohydroxyethyl terephthalic acid. Then the second enzyme is which hydrolyzes the MET into two monomers of terephthalic acid and ethylene glycol. And there are engineered Pseudomonas putida strains which can utilize ethylene glycol as a carbon source and convert it into valuable products like polyhydroxyalkanoids such as polyhydroxybutyrate, etc. Polyurethane is also widely used in the form of foams, adhesives, and medical products. Polyurethane is biodegraded through the hydrolytic cleavage of urethane bonds. Fusarium fungi and bacterial strains like Pseudomonas aeruginosa, Pseudomonas struzzi, and Commomonas stauberans are known to degrade polyester polyurethane. The degradation occurs in two steps, as these bacteria form a film on the surface of the plastic or microplastic, followed by the hydrolysis of the ester bonds. The enzymatic breakdown of PET is a very sophisticated process, and it involves three steps: initial depolymerization, the secondary processing, and the high temperature hydrolysis. The PETase initiates the process of cleaving the PET polymer chains, generating multiple products including MET, TPA, bis hydroxyethyl terephthalic acid, and ethylene glycol. The secondary processing involves the metase, which takes over the process of met intermediates, systematically converting them to the final products of ethylene glycol and terephthalic acid. Petase and petase, they have a structure which is very much similar to cutinases, And cutinases also provide an alternate pathway which is capable of hydrolyzing PET directly into TPA and EG at elevated temperatures but in very low quantities. This is the structure of the PETase from Ideonella sacaensis, and you can see the different molecules which bind to the active site of this PETase, which includes cutin, which includes triglyceride, paranitrophenol, paranitrophenyl butyrate, paranitrophenyl acetate, hydroxyethyl methyl terephthalate. All of these are not actual substrates, but they can actually bind the active site of the PETase of Ideonella. So people have overlaid or superimposed PETase with other homologous enzymes such as cutinase of different organisms and lipase of other organisms. For example, if you can look at all these structures, Streptomyces exfoliatus lipase and the leaf branch composite bacteria cutinase. The leaf branch composite bacteria cutinase is obtained from metagenomic library, and they have cloned it, and they have expressed, they have purified the 3D structure of the protein is known. And if you can see, there is a huge overlap of these petases and with the other cutinases, that is from Saccharomyces viridis and other Thermobifidia fucosa cutinases. So if they are very similar, do these cutinases also have similar activity as PETase, or does these cutinases are as efficient or as PETases of Ideonella sacaensis? No. We will see why it is having so much better activity at degradation of PET than these cutinases when you know the overall structure of this PETase from Ideonella sacaensis. So basically, the PETase is a alpha beta hydrolase fold containing an esterase, which is having a serine histidine aspartate catalytic triad in its active site, and which is very much essential for PET hydrolysis. The structure of this enzyme enables a proper binding of the substrate, that is a polymer of PET. The surface of this PETase is a very much polarized surface with a isoelectric point of 9.41, differing from other PETase variants and polyester hydrolases. There are two disulfide bonds which contribute to the stability of this enzyme. And some of the differences with the metases, which are from the same organism, is that this petase is very small and which is very much similar as the cutinase. We will see how the size of which will have the space for the polymer to bind, and there is a serine histidine 208 and aspartate 177, which is close to the substrate binding cleft. And there is also a tryptophan 156, very close to the active site. And there is a methionine 132 and threonine 58, sorry, tyrosine 58, which are also very close to the substrate binding pocket, and which forms a oxyanion hole for the substrate to properly align. The keto group of the PET properly aligns with the oxyanion hole, and the tryptophan 156, as you can see in the left side active side, it shows various conformations. But as soon as the PET is present in the active site, the tryptophan 156 is stabilized, and it is stacking with the aromatic ring of the PET. Once they are properly aligned, the serine 131, which is the part of the catalytic triad, will do its activity, and there is a cleavage of the PET chain, and the monomer is released from the active site. As we have seen that the structures with bound substrate and product analogs, HMET and paranitrophenol, reveal ligand binding to the shallow cleft formed by the hydrophobic residues on the protein surface. The substrates interacting residues are like tryptophan 156. This 156 is very tryptophan 156 is very important, as it has a wobbling conformation. That is, it is very much easy to have different conformation, but as soon as the substrate is present in the active site, it stabilizes the interaction. There are other residues like serine 185 and other interactions, which also help in overall stabilizing of the molecules present in the active site. We'll see why it is very difficult to break PET. So you can see in these top structures, there is a semi-crystalline PET. In the central region, there is a crystalline region, and in the periphery, there are mobile amorphous regions which are easy for accessible to the enzyme pentase. But when we treat with physical treatment or like chemical pre-treatment or any other activity, so the crystalline region which is present in the center will be converted into a amorphous region, and once this is converted into an amorphous region, there is a chance of more enzymes being occupied on the PET amorphous region, and which leads to formation of these compounds like bis hydroxyethyl terephthalate or monohydroxyethyl terephthalate, terephthalic acid, and ethylene glycol. As I have told you that there is a glass transition temperature that is important that we understand this, and why thermophiles are more suited for degradation of PET rather than the cold adopted enzymes. because for bulk amorphous PET, the transition temperature is 65 to 71 degrees. When it is present along with water, it lowers the glass transition temperature up to 16 degrees due to its plasticizing effect. The surface layer of PET, if it is very thick, that is present in microplastics or nanoplastics, which is 13 nanometer thick, it has even lower temperature, like 48 degrees, and when it is present with water, it can drop below 40 degrees, making it easier for enzymes to act on it. As I told you earlier, that there is a huge difference between the size of PETase and metase, even though the monomers, the polymers which bind to these active site, they are very similar. Both enzymes share the classic alpha beta hydrolase fold and have a conserved serine histidine aspartate catalytic triad, and both hydrolase ester bonds using the same triad. But metase has an extensive lid or a bulky lid of 240 amino acid residues that encloses the active site. The reason this bulky lid is required because it has to create a hydrophobic region within the active site for it to act on the monomer MHET. The MHET lid dictates directly its substrate specificity. Any modification on the lid alters the catalytic activity of this metase. The metase forms specific hydrophobic and hydrogen bonds within MET, whereas PETase accommodates PET with its more open binding cleft. This open binding cleft is required for the huge PET plastic polymer to be accessible to the active site. Some of the other unique structural elements about metase is it has an active disulfide bond within the lid, which is absent in the PETase. The lid present in PETase is only 7 amino acids, whereas the metase has a 240 amino acid big lid. As you have seen that at elevated temperatures, there is a good chance that PET will be degraded, and normal enzymes, mesophilic enzymes and cycrophilic enzymes have less chance to work on bulky PET polymers. So thermostable enzymes, they are more stable at higher temperatures. Metal ions like calcium and magnesium also improve the enzyme stability. Many of the calcium ions bind near the enzyme active site and help regulate the opening and closing during the substrate interaction. However, there is a downside that calcium can interact with terephthalate, the breakdown product of PET, forming an insoluble byproduct and inactivating the enzyme. But we all are in need of cold adapted petases and metases. The reason being, most of the microplastics are ubiquitous in the environment, but The accumulation happens in the oceans and our river systems. As they accumulate, they form the products which are accumulated, magnified in the food chain, and ultimately lead to food and feed components of our environment. The unique conditions of these cold habitats, they necessitate specialized solutions for degradation. Some of the psychrophilic enzymes, which have an active or optimal active temperature between 0 to 20 degrees, they have unique structural flexibility which make them catalytically active at this low temperatures. Here we can see some of the bacterial cycrophiles for microplastic degradation. Some of them are Pseudomonas, Rhodococcus, Streptomyces, Bacillus, Orthobacter, Micrococcus, Corynebacterium, and some of the cold adopted species are Alcanivorans, Pseudomonas citronellosis, Bacillus flexus, which can specifically degrade PVC, and Comomonas acidovirens, which can degrade polyurethane. There are many fungal species also which can degrade this microplastic. Many of the mushrooms, such as pleurotus and many white rot fungi, Fusarium and Aspergillus niger, they can also degrade something like polypropylene or polyurethane. There is a huge gap in studying how these cold adopted enzymes differ or are similar with the mesophilic petases or metases. Further research is needed to characterize and enhance the psychrophilic enzymes for activity and efficiency of microplastic degradation in cold environments. Most of these psychrophilic cold adopted enzymes are isolated from alpine regions, Himalayan belt, Antarctic and Arctic regions. These cold adopted enzymes, which are produced by psychrophilic organisms and are involved in microplastic degradation, they are very high in catalytic efficiency at low temperatures and enhanced structural flexibility and modified active sites that facilitate substrate binding and reaction at suboptimal temperatures. So, why do they have structural flexibility? These organisms have structural flexibility, or these enzymes from cycrophiles have structural flexibility because they have weak hydrogen bonds and weak salt bridges, whereas thermophiles have strong salt bridges and strong hydrogen bonds, so they are very stable at higher temperatures. The active site of most of the cycrophilic enzymes are large and increased hydrophobic nature. The reason is they have large loops which can accommodate large molecules because at low temperatures it is a need for them to accept molecules um, because of the thermodynamically not stable conditions. amino acid compositions of most of the cyclophilic enzymes have very less proline, and they are very high in glycine, methionine, and asparagine. But the downside of these enzymes is that these enzymes are only stable at very low temperatures, but they are not useful for any mesophilic. And they are able to protect this the solute production. Because of this, they have adapted membranes with high fluidity. Apart from the petases, there are other cold adapted enzymes which are involved in microplastic degradation, such as esterases, lipases, and cutinase variants, which are isolated from many cyclophilic bacteria and fungi found in polar, alpine, and deep sea environment. As we have seen the structure of the petase from Adenola sacchaensis, that it is a alpha beta fold containing enzyme with which a triad of serine, histidine, and aspartate. And most of these enzymes also have a alpha beta fold and with the same triad, but the crystal structure of the or overall structure of these enzymes is quite different. For example, lipases from cyclophilic organisms like Antarctica, Pseudo Alteromonas, and Pseudomonas species remain highly active at 0 to 15 degrees, contributing to hydrolytic breakdown of various synthetic polymers. Similarly, cutinases or cutinase-like enzymes from Spingomonas, which are adapted for cold climate, have displayed activity on aliphatic polyesters like polycaprolactone at low temperatures. Now we will compare some of the pet hydrolysis from marine microbes with petase from Idenella sacchaensis. Some of the enzymes which are pet hydrolysis, they are named based on the organism, like Oleospira antarctica. We call the petase as PET5 or OA cute meaning the cutinase kind of structure. Moraxella species, the petase is called as MORS1, and similarly there are many other petases which are given a name like PET6 or PET6, and the variants are named based on the variant where the variant is or the mutation is located. Some of the PET hydrolyses are derived from the metagenome. They are derived from metagenomic library. They are cloned and expressed in organisms like E. coli or yeast, and their activity is studied, the structure is studied, and some of these enzymes are named as Ple628 or Ple629. Some of the structural features of these marine cold adapted petases is they have a very optimal temperature at 0 to 20, but there are certain exceptions. Some marine organisms have petases which have an activity at higher temperatures also. For example, PET 46 shows a high TM at 84.5 degrees centigrade, allowing function at 60 degrees, making it as a promising for high temperature industrial recycling. Most of these petases, they have two disulfide bridges as the petase of Ideonella, but some of the enzymes of PET6 or MORIS1 have a rare third deep disulfide bridge, which is similar to metase of Ideonella sacchensis, which is helping them to stabilize for cold conditions. Most of the petases they have from the marine organisms have very high tolerance for salinity, and they are usually stabilized by sodium chloride. Some are very much stable at zero point 0.5 molar NaCl, and some are more stable at 1.5 molar NaCl. PET 46 and MT-cute, they have an active site which prefers crystalline or semi-crystalline PET over the amorphous PET. So these enzymes are also very much useful for scientists, where they can use these enzymes for initial breakdown of the crystalline PET into amorphous, which will be later uh, used by petases and metases Ideonella, because they prefer amorphous nature of the PET. Most of the PET, as we have seen, the Ideonella species of PETase as having a very short lid. The metases is having a very bulky lid of 240 amino acids, but the petases from the cold adopted or from the saccharophytes has a medium sized amino acid lid of 45 amino acids, and sometimes they have a 70 amino acid lids, and they also have other activities such as the endo and exoesterase cleavage actions within the lid. They also have multiple substrate binding domains. That means it can incorporate or accommodate more substrates than petase of Ideonella kinensis. If you want to make an enzyme which is very much useful for industrial application, where you want to make the best plastic degrading enzyme, especially microplastic for industrial application, then we need help of both enzymes which are similar to Ideonella, and we need metases some much similar to Ideonella, and enzymes such as cutinases from Pseudomonas rhodococcus, and also the other enzymes like lipase structure containing enzymes and these variants. Some of the variants I have shown you here, which have shown exceptional activity. The first variant is PET with a histidine variant or a tyrosine variant, which is converted into serine in the active uh, site cleft region, where the tyrosine is changed to a smaller moiety serine. It has increased the activity by 1.3 fold because it is more accessible for the semi-crystalline PET. PET6 with a external loop or extended loop region. The replacement of the external extended loop region with IS PETase sequence, that is the 7 amino acid residue of Iodella sacchensis PETase. It outperformed wild type PET6 by 79% in hydrolysis product released from post consumer PET at 50 degrees centigrade and 1 molar NaCl. So basically, what they have done is they have taken the ideal features of the PETase from Iodella sacchensis and incorporated in the PET6 enzyme, which is a marine psychrophilic enzyme. Because of the exception is that even though it is a psychrophilic enzyme, cold adapted, it is also stable at very high temperatures. So we can use it at 50 degrees centigrade and at, at a high salinity of 1 molar NaCl. There is another mutant which is called as PET6 VSAT. There are mutations in the loop regions where the valine at the position 91 is converted into threonine and the serine is converted to alanine. So it outperformed the wild type PET6 by 58% and iodokinin Iodella sacchensis PETase by 1.6 fold in hydrolysis product. It is released at 50 degrees centigrade and 1 molar NaCl. So we can actually mix the better mutants or better features from other proteases and create chimeric enzymes and make useful enzymes which are tailored for different conditions. For example, if you want to generate a mutant which is used to degrade PET at coastal regions where the salinity is more, then we can take the help of sacrophiles and use their petases and incorporate the best mutations from the mesophilic Iodella sacchensis and create a chimeric enzyme and degrade PET. And in the same way, or vice versa, we can actually create a petase which is more stable by incorporating thermophilic features such as salt bridges or more hydrogen bonding to increase the thermal stability of the petase of Iodella sacchensis so that it can be used at elevated temperatures like 41 to 60 so that even at mesophilic conditions or at higher temperatures, more of the plastic can be degraded. I thank you, one and all, for giving your attention. And if you have any questions and if there are any discussion points, and I'm willing.
Uh,
Sir, Pal sir,
Yes.
is it over?
Yes.
First, first session?
Yes, madam.
Yes.
Yes.
Thank you, sir. Let me give vote of thanks. Take a few minutes, then again second session will start. Okay, sir.
Thank you
Ma'am,
very much.
and one more thing, sir. Somebody shared questions, some questions. They got doubts regarding few questions, 3 questions.
Yeah,
Sir, what are the major classes of enzymes involved in plastic degradation?
major classes are hydrolase. The major class is hydrolase, and if you can subdivide them, we can call them as cutinases or esterases or lipases. All of them are having a alpha beta hydrolase fold. So there are many enzymes with the alpha beta hydrolase fold, but in the enzymes which are having alpha beta hydrolase fold, this triad of serine, histidine, and aspartate should be present so that it can have some activity.
Uh,
So
and
when they have this kind of futures, there is a possibility that it can have some activity. Let me explain. I have shown you some of the these molecules like paranitrophenyl acetate, paranitropbutyrate, or paranitrophenol. If they are able to bind in the active site, then there is a chance that these alpha beta hydrolase containing enzymes might have the ability to degrade PET.
so one more question, sir. What what are the
Yes,
factors considered for measuring enzyme stability?
can you please repeat?
What are the factors considered for measuring enzyme stability?
So, what are the factors considered for enzyme stability? Enzyme stability is like is the enzyme thermally stable? That is first question. Second question is the enzyme stable uh, for exposure to solvents? So, and if I ask these questions, then they are quite different. So, for thermal stability, we need high salt bridges and very high hydrogen bonding. So, if you want to make a mesophilic enzyme to make it into a thermal stable enzyme, we should try to stabilize by very high hydrogen bonding, preparation or incorporation of salt bridges, and sometimes we can also incorporate disulfide bonds within the molecule to stabilize the overall structure of the enzyme. Sometimes um, it is not even possible to incorporate disulfide bonds in any molecule. It might be deleterious for the stability also or activity.
So
I hope I answered that question.
one more question, sir. Can we use enzymes present different food stuff for degradation?
Can we use?
Enzymes present different foodstuffs for degradation.
I did not understand the question. Can we use enzymes for degradation of food? Is that the question?
Yeah, sir. Maybe related. Many questions are
So,
there.
usually, if there is spoilage of food, that is because of the enzymes only produced by different organisms like amylases, lipases, or esterases. So all these will degrade the food. And if there is any specific thing I can answer about this, they can ask a continued question on that.
Is there sensors to detect microplastics in water?
That we can um, talk in the next session, because I'm going to talk about detection of microplastics.
So can
And
enzymes be tailored to degrade multi-layered or mixed plastics? Can enzymes be tailored to degrade multi-layered or mixed plastics?
so it's very difficult to degrade multi-layered plastics. Multi-layered, even multi-layered in the sense enzymes like petases, they are only working on amorphous form of the PET, the polymer. It is not attacking the crystalline form of the PET, which means that when the plastic is very much compact, like multi-layered, it is not possible to use enzyme for degradation. We need a physical form or a chemical form to actually break it, so that the amorphous form is available for the enzyme. And the other second part of the question is, if there are mixed polymers, can we degrade? A single enzyme can have activity on one type of polymer. So, if you want to degrade a mixed polymer, then you need more enzymes or different types of enzymes to degrade these polymers. For example, alkane hydroxylases are required for polypropylene or polyethylene kind of polymer degradation. Whereas for PET, we need petases or cutinases or lacases kind of enzymes for degradation of PET. Polyurethane, we also need different kind of lacases, which are like uh, dioxygenases or monoxygenases, which attack these polymers, oxidize, and then they can degrade. So for mixed polymers, you need different types of enzymes, not one single type of enzyme.
Okay, sir, let me ask two more questions. How can CRISPR technology contribute to the development of efficient plastic degrading microbes?
The advantage with CRISPR is you can incorporate any mutation or incorporate any gene in the desired location on the genome of an organism without uh, changing much of the genome. That means you don't have to have an extra plasmid, or you don't have to introduce other mutations or scar regions in the genome. Uh, if we are able to select the gene, we should be able to incorporate a particular type of mutation or gene in the organism without any scar or without any additional sequences. So you will not be changing the fitness of the microbe. For example, in E. coli, if you put a plasmid, um, there is a chance that its fitness decreases because of the burden it has to multiply the plasmid. And so if it is incorporated in the genome, it is a single copy on the genome. So it is very easy, that it is stable, it is not lost while replication. So these are some of the advantages with CRISPR.
So one more question, sir. I think it may be last question. How can molecular docking be sorry? How can molecular docking be used to study enzyme plastic interactions?
Um, See, for this plastic degradation,
Yes.
some of the
Docking.
structures are well known. Yes. We need a PDB structure or a structure which you have generated uh, to initially start, um, or initially you have to have a 3D structure so that you can dock this um, polymer and find what are the interactions in the active site. You can see how it is interacting with the other residues in the active site, and you can see whether there are the whether if the catalytic amino acids are properly oriented or not. So all these can be studied using docking, but you need the three-dimensional structure of the protein first uh, to study.
Sir, one more question. What is the commercial availability status of these enzymes?
I don't think these enzymes are commercially available, uh, but most of the research groups they are actually working on making these enzymes.
So, any more questions? So, thank you, sir. Thank you for your valuable speech for the session one, day one. Okay, take a few minutes. Again, we will start at 8 o'clock. Meanwhile, I will give vote of thanks for session one.
Thank you.
So, dear participants, I think I hope you will enjoy this session. So, I extend my heartfelt gratitude to Dr. Emmanuel Paul, head of the Microbiology and Biocatalysis, Zanoprima India Private Limited, for his inspiring lecture on the role of cold adapted enzymes in microplastic degradation. Your 20 years of expertise and clarity of vision enriched our understanding of microbial biocatalysis. I also thank the participants for their active and thoughtful engagement and the way of questioning. Thank you, one and all, for this session one. Again, we will join at 8 o'clock. So, meanwhile, I want to give some suggestions for participants. So, regarding use name which is appeared in the Zoom meeting, somebody has some number, somebody has some mobile number like Samsung or Xenoma, something like that. So kindly ensure that make your username or whatever the register name you can make it. You can revise with the number or this mobile name you can change with your name. that is better to take attendance. We will post attendance at the end of the session. Sorry, I think it was muted. Now I unmuted. Dear participants, please ensure that you can join with us with your register name instead of number or a mobile name, etc. So that is easy to take attendance for us. So every day we will post the attendance. So make it clear to avoid